Hello everyone, welcome to our video. In today's video, we will be discussing about a food poisoning outbreak that happened after a food fair at a school. In today's video, we will be discussing about the introduction to the case study presentation, diagnosis and tests done, prevention and control measures, conclusion, and a list of references. Here's the introduction to the outbreak. A food fair was held at SMK Jalan Melor in conjunction with the school's annual co-curricular week. The various vendors were invited to set up stalls selling food and beverages including fried chicken, fried noodles, fried rice, chicken rice, sandwiches, beverages, and many more. After the event ended, multiple students developed gastrointestinal symptoms within hours after consuming food from the fair. The symptoms included nausea, vomiting, abdominal cramps, and diarrhea. After the fair ended, 5 of the fair attendees showed up at the clinic after showing symptoms. Patient A, 16 years old, had fried rice and ice drinks. Patient A had repeated vomiting and abdominal pain and fever. These symptoms showed up after 4 to 5 hours consuming the food from the fair. Patient B, on the other hand, 17 years old, had fried rice, fried chicken, and bottled water. This patient showed up to the clinic with nausea, vomiting, and mild diarrhea, and no fever also. The symptoms showed up 6 to 8 hours after the fair. Patient C, 15 years old, had sandwiches, packaged snacks with mild abdominal discomfort after consuming the food, and there is no fever also. Patient D, on the other hand, 16 years old, had fried noodles, ice drinks, and showed up to the clinic after mild nausea. Also no fever, and the onset was 10 to 12 hours after consuming the food. Last but not least, patient E, 17 years old, only had fried rice and had severe vomiting, abdominal cramps, and no fever. The symptoms showed up 3 to 4 hours after the fair. From the patient profile shown previously, we can identify that the common food link that led to the symptoms is fried rice, as seen in patient A, B, and E. All patients showed to the clinic without fever. Incubation period for the symptoms to show is around 3 to 12 hours. Some patients showed dehydration due to diarrhea and vomiting, but most of them are stable. Our suspected diagnosis for the causative pathogen in this case is the emetic form of Bacillus cereus, Staphylococcus aureus, or Clostridium perfringens.
Moving on to our differential diagnosis, our strongest etiological match for the acute cases in this outbreak is actually Bacillus cereus, specifically the emetic type. When we look at the epidemiological data here, there's a clear pattern matching our food wiggle and clinical timeline. Starchy foods, classically the fried rice consumed by multiple students, provide an ideal environment for B. cereus spores to survive cooking and germinate during temperature abuse and produce a heat-stable toxin called as cereulite. And what makes this as our primary suspect is the hyperacute onset window of 1 to 6 hours. This perfectly accounts for patient A and as well as patient E, who both fell violently ill within 3 to 5 hours of eating. From a physiological standpoint, the ingested cereulite toxin will actually directly stimulate the vagus nerve in the duodenum and sending rapid signals to brain vomiting center. And it will eventually trigger severe and repeated emesis and as well as abdominal cramps, characteristically without fever, which is perfectly mirrors our cohort's presentation. Now, we cannot look at Bacillus cereus without immediately considering our second differential, which is the Staphylococcus aureus enterotoxemia. This is a highly possible alternative or concurrent cause for several reasons, because S. aureus contamination typically stems from a poor hygiene during manual food preparation. And given that this school was having a food fair with multiple stalls handling items like sandwiches and as well as the fried chicken, and therefore manual handling was undoubtedly extensive in this event. In medicine, we describe these two conditions as clinically indistinguishable because both pathogens involved in preformed heat-stable toxins acting on nearly identical pathways, and they both cause a rapid incubation period of 1 to 8 hours, a massive focus on vomiting, and a notable lack of fever. And therefore, without laboratory isolation and toxin assays from the food odometers, it is virtually impossible for us to separate them based on symptoms alone. And this makes the S. aureus as a strong suspect, particularly for patient B, who consume both fried rice and fried chicken. However, a single pathogen toxic exposure doesn't fully tell the whole story. To achieve an accurate diagnosis, we must address the 10 to 12 hours cohort. And based on this case, not all patients fell ill immediately with acute vomiting. As noted in our case profile, patient D and several other students only began feeling unwell late overnight or the following morning. And their symptoms were completely different, as some of them actually experienced mild nausea and prominent abdominal cramps and diarrhea, but no vomiting at all. And this distinct clinical picture and delayed timeline point directly to Clostridium perfringens. And sea pathogens require a longer incubation window because it relies on the ingestion of vegetative cells that multiply and produce enterotoxin in vivo within the small intestine. And therefore, our scientific conclusion is that this school fairly experience a dual pathogen exposure rather than a single compromised source. The data also strongly suggests that the temperature abuses occur across multiple independent food stores, causing an acute emetic outbreak during the day, followed by a secondary diarrhea wave overnight.
In this outbreak, Bacillus cereus can cause two different types of illness. The first is the emetic type. People eat food that already contains the toxin. The toxin is called cereulide. It affects the stomach and the vagus nerve, so patients vomit very quickly, usually within 1 to 6 hours. This matches patient A and E. The second is the diarrhea type. The bacteria grow inside the intestine and produce toxin there. This toxin damages intestinal cells and cause diarrhea and stomach cramps after about 8 to 16 hours. This is similar to patient D.
Now I will be talking about the diagnostic methods of this case study. Firstly, we need to collect samples from the patients and the environment. This includes stool and vomiter samples from students with symptoms, leftover fried rice because it is the suspected food source, and swabs from the food preparation surfaces and utensils. Next, laboratory tests are carried out. If the bacteria is Bacillus cereus, the gram stain will show large gram-positive rods. On MYB agar, it produces pink colonies with an opaque precipitation zone. To rule out other food poisoning bacteria, Staphylococcus aureus appears as gram-positive cocci in clusters and form black shiny colonies on Burke-Parker agar. Clostridium perfringens appears as large gram-positive boxcar-shaped rods and form black colony on TSC agar under anaerobic conditions. These results help confirm that Bacillus cereus is most likely cause of this outbreak. Now I'll be continue on the treatment plans of this case study. Treatment for this type of food poisoning is mainly supportive because it is caused by cereulide toxin, not by an active bacterial infection. For patients with mild symptoms, oral rehydration solution is given to replace the fluid and electrolyte loss through vomiting. Patients are also advised to rest and drink enough water. If the patient becomes dehydrated, intravenous fluids are given. Antiemetic medicines can also be used to reduce nausea and vomiting. Although vancomycin is an effective antibiotic against Bacillus cereus infections, it is not recommended in this case. This is because antibiotic cannot remove the cereulide toxin that has already been eaten. Most patients recover within 24 hours with supportive care only.
Here comes the prevention and control measures. Firstly, food hygiene and preparation. We need to ensure that we cook the food truthfully, 75 degrees Celsius or above for poultry, 65 degrees Celsius or above for rice and noodles. Also, we need to avoid leaving cooked rice and noodles at room temperature to avoid Bacillus cereus toxin. Secondly, storage and handling. We need to refrigerate the perishable food like salads or fruits until serving. Also, we need to discard the leftover food that are not being consumed within 2 hours. Thirdly, training and supervision. We need to train the F&B workers in hygiene, make sure that they are always washing their hands, wear the glove and hairnets before processing the food. And also, we can require more food safety certification for vendors. Last but not least, clinical and public health measures. We need to let public know that how to recognize the symptoms early so they can refer to supportive care early. Also, report suspected outbreaks to health authorities for lab testing or further medical.
In conclusion, this case shows that it is important to look at all the information before making a diagnosis. These symptoms start with very quickly after eating, and the laboratory finding also help us understanding what happened. After comparing different bacteria, Bacillus cereus is the most likely cause of this food poisoning. Most patients can recover well with enough fluid, rest, and medicine to control the symptoms. Antibiotics are usually not needed because the illness is caused by a toxin in the food. This case also reminded us that good food hygiene, safe food storage, and proper cooking are very important to help prevent food poisoning in the future.
As the group leader for this assignment, my main role was structuring our content, guiding meetings and ensuring everyone contributed fairly to the assignment. Our team's strength was organizing the information provided into a logical sequence together, and this made our case analysis clear and engaging. I also focused on keeping the team motivated and on track. The biggest challenge was coordinating different schedules and managing disagreements in the beginning of the assignment, especially when some of them leaned towards Staphylococcus aureus infection, while others argued for Bacillus cereus. We all did our own research, and alongside, I facilitated my members with evidence-based discussion, ensuring everyone's voices were heard. We presented our findings to each other in discussion and came down to one conclusion. The experience taught me that effective leadership isn't about having all the answers, it's about creating an environment where the team can find solutions together. Moving forward, I'll apply these teamwork and communication skills in clinical settings where collaborative decision-making directly impacts the patient care.
This presentation helped me learn more about Bacillus cereus food poisoning and how it is diagnosed and treated. My main contribution was preparing the diagnostic method and also treatment plan of this case study. I also searched for a journal article to make sure that the information was accurate. Working with my teammate helped us complete the presentation smoothly. One of the challenges that I faced was understanding why antibiotics are not used to treat this type of food poisoning. At first, I thought all bacterial diseases should be treated with antibiotics. After reading more journal articles, I learned that this illness is caused by a toxin, not an active bacterial infection. This helped me understand the topic better and improve my critical thinking. In the future, I hope to improve my presentation skills and apply what I have learned in microbiology studies in my future career.
During this project, I learned many new things about food poisoning and how to solve a real case. My job was to prepare the pathophysiology and conclusion. I researched for information, read journal articles, and worked with my teammates to make sure our answers were correct. The biggest challenge for me was understanding the medical terms and reading English articles. Sometimes it was difficult, but I asked my teammates for help and spent more time learning by myself. For this project, I improved my research skill, teamwork, and communication skills. In the future, I will continue to improve my medical knowledge and use this skill in my studies.
In this assignment, my role was to investigate the differential diagnosis by researching all possible pathogens that cause the listed symptoms. I identified the top 3 likely causative pathogens, which was the Bacillus cereus, Staphylococcus aureus, and Clostridium perfringens, and proposed them to the team, ensuring that we reach a mutual consensus. However, this project would not have run smoothly without my group members, who utilized excellent organization skill to synthesize our cooperative diagnostic metrics. Our primary challenge was managing the clinical uncertainty surrounding the overlapping timelines of Bacillus cereus and as well as Staphylococcus aureus. Initially, our thinking was limited to finding a single source. However, by engaging in structured team sharing and synthesis, we managed our conflicting viewpoints objectively. Our mindset eventually shifted entirely and leading us to recognize the data and actually pointed to a more complex dual pathogen exposure across multiple food stores. This assignment fostered significant personal and team growth, particularly in evaluating epidemiological data critically. And looking ahead, an area for improvement is actually refining the speed of our initial diagnostic differentiation. And cultivating this strong analytical insight provides a direct and more meaningful connection to my professional development and as well as future clinical microbiology practice.
So for our case, I handled the prevention and the control side. Honestly, I didn't just want to list the textbook's rules like wash your hand and keep the food cold. I actually go through more and more articles and a real case study to figure out what actually go wrong in the kitchen. This part really felt very good for me and I enjoyed it very much. What tripped me up was the amount of information out there. There are so many guidelines and I keep going and thinking, am I including too much and I did missing something I need to include in a slide? By going through that, this project really inspired me a lot on my understanding on microbiology in food.